In today's program, we will deal with the protection of our crops. We will then see how plants are screened for disease. And finally, we will look at various transformations undergone by another food product, milk. Nowadays, industrial-type farming holds sway. Farms have become veritable commercial enterprises. In some respects, the planet demands it. Every year, there are 80 million new mouths to feed. If we succeed to some degree, it's largely due to the development of new farming techniques. But there is a price to be paid. In the past 40 years, the amount of pesticides and fertilizers used has soared. Excessive use of these products is not always effective, and it can harm the environment. Fortunately, today more refined means are made available to farmers. Ever since man began working the land, he has had to fight a constant battle against all sorts of organisms that attack his crops. Insect and mite pests, rodents, pathogenic organisms, and weeds can often endanger crops and affect the quality of our food products. To counter these scourges, producers resort to various methods, among them the application of pesticides. We know that these products are potentially harmful to the health of humans and contribute to the pollution of our environment. That's why agricultural research is developing and refining integrated pest management programs. The programs are based on the rational use of pesticides and the promotion of biological and mechanical pest control. So protecting crops is now based upon new technologies, especially in the area of forecasting infestations and diagnosing pest problems. Bioclimatology enables scientists to follow the evolution of such biological phenomena as certain pathogenic insects or fungi. Bioclimatology is the analysis of data collected on a daily basis in small weather stations and biological observation posts located at strategic points on the farmland. By compiling the data on temperature, precipitation, and humidity from the various weather stations, scientists have managed to put together data banks on the history of certain biological events. These data banks have enabled scientists to identify risk areas and to develop forecasting models. Thanks to these models, the researchers are able to identify with a fair degree of accuracy the periods when a disease or an insect pest invasion are most likely to occur. For example, we know that X number of hot, dry days can cause an increase in the type of mites that attack crops, including apples and raspberries. The climatic data is rounded out by biological information obtained through observation and trapping networks, which detect any sudden increase in a pest population. Trapping is done with traps filled with synthetic sexual hormones, pheromones, or a food, or simply a color lure. Pheromones are odorous substances that are naturally secreted by the females of certain insect groups during mating time. Pheromones are specific to every species of insect. The traps are used to detect apple tree insect pests and corn moths. When the data has been analyzed, a pest warning is sent out by the Pest Control Warning Network to the farm producers and agricultural representatives. The warning outlines the pest situation for the crops in every region and explains what to do in each case. Of course, insects and diseases are not the only enemies of crop plants. Farm producers also have to contend with any number of varieties of weeds that give their commercial crops a hard time. Sharing the same lot, the two types of plant often compete for nutrients and light, which naturally affect the yield. For these plants, the growing field becomes a battlefield. So one of the greatest challenges modern agriculture faces is still locating and getting rid of weeds. To keep the use of insecticides down to a minimum and use only the appropriate ones, the farmer has to be able to identify the weeds in their early stages of growth. This is easier said than done, since in the early stages, the plantlets look a lot alike, which makes identification virtually impossible, even for a seasoned farm consultant. Fortunately, computers have recently come to the producer's aid. A software program has been designed to help identify the pest plants as soon as they emerge. The knowledge and know-how of specialists in their field are translated into an expert systems software program. Through a series of questions and illustrations, the computer lets the user recognize a sample accurately in about 10 minutes. The appropriate treatment can then be suggested. While the expert system is in itself complex, using it is relatively simple, even for a neophyte. The computer asks a question and displays an assortment of pictures as possible answers. All the user has to do is select the picture that matches the sample. Applying these new technologies may pave the way to more productive farming, capable of feeding the world without undue damage to the environment. Present-day farming is changing. There is a burgeoning interest in mixed crops. Cultivating several varieties of plants in the same field prevents the soil from becoming depleted. If several rows of grains are planted alternately with several rows of legumes, the latter, which fix nitrogen, keep the soil fertile. This provides an advantage for the grains which do not have that property. The new technologies all contribute towards bringing about the model farm. Genetics, for example, has helped develop more resistant or better adapted strains. It is also a valuable tool in the area of diagnosis. In plant protection, as in medicine, it's important to make an early and accurate diagnosis in order to prescribe the adequate response or medication, and also to avoid irreparable damage. For a long time, scientists trying to identify pathogenic organisms have relied upon a battery of biochemical and biological tests and made observations with photonic and electronic microscopes. However, the process was often long, costly, and was not always as precise as could have been desired. In recent years, however, the introduction of immunological means has helped to speed up diagnoses and make them more accurate. The serological means most used worldwide is the Elisa Assay, a test used both for humans and plants. An extract of the plant is placed in the presence of an antibody to detect the presence of a pathogenic organism. The antibody is taken from an animal, usually a rabbit, in which the suspected pathogen is introduced. In response to the virus, the animal produces specific antibodies that are collected by taking a blood sample. The blood is then broken down to obtain the antibody. Because a plant virus is involved, the procedure does not harm the animal in any way. In the laboratory, the antibodies are attached to the walls of cavities in a plastic plate. An extract of crushed plant is placed in them. The microbes in an extract of an infected plant will be retained by the antibodies. Another drop of antibodies bonded to an enzyme is added, after which the cavities are washed. Only the cavities containing the microbes will hold the enzyme. When detected, the enzyme produces a yellowish color. This means that the extract of crushed plant had microbes in it, and this confirms the presence of the disease looked for. Also, the intensity of the color indicates the quantity of pathogenic microorganisms in the sample. Increasingly sophisticated, biotechnology is revolutionizing how we screen plants for disease. For several years now, researchers have been working with nucleic probes capable of recognizing genetic sequences exclusive to certain pathogenic microorganisms. Every living organism has a genetic heritage that determines its size, shape, and color. That heritage is inscribed in the deoxyribonucleic acid, or DNA. DNA is made up of two complementary strands bonded together by nucleotides, the base substances of all organisms. The nucleotides, of which there are 4 types, are bonded in pairs according to a precise rule. Thymine always bonds with adenine, and guanine with cytosine. We call a specific sequence of nucleotides a gene. Indeed, in the DNA of a given species, there are nucleotide sequences that are exclusive to that species. A nucleic probe is a fragment of DNA whose nucleotide sequence is complementary to a nucleotide sequence which is exclusive to the microbe looked for. The length of a probe can vary between 20 and 5,000 nucleotides. To make a diagnosis using the nucleic probe technique, the researcher first extracts all the DNA from the tissues of the plant believed to be infected. The 2 strands of DNA are separated. The DNA strands are then mixed with a solution containing several million copies of the nucleic probe. If the disease is present, the probe will pair up with its complementary sequence contained in the microbe's DNA. However, the probe will not be able to pair up with any other sequence of nucleotides from the plant or any other organism. Since the pairing of the DNA probe and the DNA is difficult to see, even under a microscope, the researchers put a marker on the probe to be able to spot the pairing reaction. The marker can, for instance, be a radioisotope, found by detecting the radioactivity. Non-radioactive markers, such as enzymes, are also used. Pairing the nucleic probes with their complementary sequence requires the total recognition of a great many points of contact, whereas the pairing of the antibodies with their antigenes only requires a few. Nucleic probes therefore offer greater diagnostic precision than techniques based on antibodies. Other diagnostic techniques are currently being explored. Genetics is helping agronomy make as much headway as it is for medicine. If we can transform seeds, then we can transform foods. Take milk, for instance. Mother's milk is the newborn's first food. But through his lifetime, man continues to consume dairy products. Over 2/3 of the world milk production is used to manufacture byproducts: yogurt, ice cream, butter, cheese. Let's take a look at some of these processes. Leading-edge technologies have not only been implemented in agriculture, but also in the food processing industry. The consumer often has the impression that the processing his food goes through removes all its nutritional value. In fact, it's quite the opposite. Automation has improved hygiene, and this has considerably reduced the risk of food contamination, which is the cause of many poisonings. Moreover, thanks to advances in technology, the experts are able to add nutritional value to foods and lengthen their lifetimes. For instance, a whole range of products can be produced from a single food. The dairy industry is one of the sectors to have most benefited from recent technology in this respect. Today, milk is the object of a veritable cracking, yielding several distinct products, a variety of cheeses, yogurt, beauty products, and even drugs. After milking, first the whole milk is screened for antibiotics. The antibiotics administered to sick cows are a health threat for humans, which precludes the milk from being processed any further. In most Western countries, milk is pasteurized. Pasteurization is a simple process that consists in heating the milk to a temperature higher or equal to 72 degrees Celsius for 16 seconds. This eliminates any microorganisms that could cause food poisoning. Any remaining microorganisms pose no health threat, but they can affect the length of time milk will stay fresh. That's why milk should be refrigerated. A sterile milk can also be made, which requires no refrigeration, based on the same process but at much higher temperatures. Requiring a far more complex heating setup, this method brings the milk to a temperature of 130 degrees Celsius for 1 second, thereby destroying any remaining microorganisms. The milk is skimmed off by centrifugation. The fat, less dense, goes toward the center of the device where it can be easily extracted. It then needs only to be remixed with the milk to obtain the desired percentage of fat content, 3%, 2%, or skim. Homogenization is an additional process whereby a pressure of over 1,000 kilograms per square inch is applied. This process causes the beads of fat to burst and prevents cream from forming on the surface. Milk is 5% lactose, an allergenic sugar that a number of people are able to digest thanks to an enzyme called lactase. This natural enzyme breaks up the chain of glucose and galactose that makes up lactose. Some groups of people, especially among black people, do not have enough of this enzyme in their organism, and therefore have violent allergic reactions if they drink milk. However, through biotechnology, a lactase can now be produced in the laboratory that rids the milk of lactose, so everyone can enjoy milk. Fermenting techniques have given us a broad range of dairy products whose nutritional values are well recognized. Yogurt is the most famous. Yogurt is obtained from introducing a bacterial culture that multiplies. Just recently, new bacteria are being used to make yogurt, such as the Bifidobacterium bifidum, that defends the human organism by protecting the digestive tract from certain infections. Cheese making has also made the most of leading-edge technology. To make milk curdle, rennet is added to it. This produces a yellowish liquid, lactoserum, that forms on the surface of the solid matter. This protein-rich liquid used to be skimmed off by hand before the cheese was put in its mold. Today, that serum is removed when the milk is still in liquid form by a process called ultrafiltration. Ultrafiltration eliminates the chances of contamination and preserves almost all the proteins which would otherwise be lost. The process uses a very fine synthetic membrane to retain the elements required to make the cheese and lets the others pass through. It works a little like a kidney filtering blood. Technological progress has not only given us cows capable of producing stupendous quantities of milk, but has also let us explore the many possibilities of that food. Milk and its byproducts have thus found a great many outlets in food, medicine, and pharmacology. Biological farming is increasingly popular, but it's not new. What do you think farmers did before they had chemical fertilizers? They rotated their crops. 2 years of grain crops, 1 year of leguminous plants, and a year of rest. They used natural fertilizers. Manure, despite a few drawbacks, is a perfectly good fertilizer.